Good afternoon. On behalf of Dr. Sinnott, welcome to Intro Medicine Grand Rounds. Today we're lucky to have one of our own, Dr. Toney. I'm sure many of you know John has been here for quite a while now, actually. I think 35 years, 36 years?
One.
Yeah. John is in infectious disease. His titles are always intriguing. You never know exactly what he's going to cover, so this is more of the same. As up there, he's been involved in epidemiology for many years. He was heavily involved with the HIV/AIDS training in the Caribbean and been involved with heading up research at the VA. And so it'll be very interesting to hear more about the upcoming sixth extinction. Thank you, John.
Thank you. Yeah, I came here in '81 to do my residency because it was certainly warmer than where I was. And I've had the best mentors, Dr. Goldman, all kinds of people who are still here, Dr. Sinnott, who would chide me as a resident. But it was one of the reasons I went into ID. And plus, when I was in med school, one of my mentors there was Dr. Maurice Mutson, who was the founding chair of medicine at Marshall. We were the first class out of Marshall for the medical school. So it's been a great relocation and great time here. And without further ado, the only disclosure that I will tell you that I have is I'm part of a Sanofi Pasteur C. diff toxin vaccine trial. It has no impact whatsoever with this. I received no money for it. It all goes to the VA research. So that's the only thing that I would disclose to you. Well, you saw in the title, I had to get something to get people's attention. It was, and I will tell you why, because when I was a resident, and it was Dr. Matt Nathan, who went on to become an admiral in the Navy and Surgeon General for 3 years up until 2015. He told me one morning in Morning Report, I said, so what's presenting? He said, I got a fascinating case of Hibgia. I said, well, what the heck is Hibgia? And he goes, come to Morning Report and find out. And we were used to seeing esophageal bleeders and all kinds of different things, kind of the more mundane routine things that we deal with. And so I go and I expect to hear this unusual thing called Hibgia. He presented a case of a guy who had cirrhosis, esophageal varices that bled. So afterward I said, well, what was that? That's the same old stuff. What's this Hibgia stuff? He goes, oh, Hibgia. Had it before, got it again. So you see this in medicine. Well, it got me interested. And this got me interested because of what you'll see as we go along. So the first thing was extinction. Most of you know the definition, but this is the definition. So it's either one of the first two, or if you're in psychology or psychiatry, it is the process of eliminating or reducing a conditioned response by not reinforcing it. Something that we wish the media might do or not do in some of these times. This is what we live on as we know it now. And this is that wonderful picture taken many, many years ago and called the Big Blue Marble. But it wasn't always like this, as most of you know, many, many years ago. It was new and it was evolving. It was mainly molten rock and to some degree metals. And there was no life on it about 4 billion years ago. But as things evolved and things cooled and we got water and we had the right time and the right place for life to come about, life did come about, and what is described as 5 past extinctions. And these are just listed for you. The first one was about 444 million years ago, and that was the Ordovician. And one of the fossils that was represented at that time, because there was a huge amount of growth apparently, and it's hard to see, but these fan-like little structures that you see at the top, they were called graptolites. About 375 million years ago, in the late Devonian period, another one, and the representative organism there was what you see as a trilobite over on the right. Tabulate coral was very common until at the end of the Permian period, where there's another extinction. Obviously, there's lots of different things that were around, but these are some of the things people note that definitely were lost. And you see the numbers on the side that are pretty impressive. Anywhere from 86 to 96% of species lost in the Permian. The one we hear about, and there were movies about the Triassic area, it was about 200 million years ago. And there was an eel-like creature that you see down here in green, and what was left fossilized was these rows of small teeth that even went down into the esophagus that were interesting. And those were the conodonts. And then the last one was about 66 million years ago in the Cretaceous period. And what you see is a nautilus-like looking object down on the bottom. That's a fossil, and those were ammonites. So we're closely related, but predated what we see now with the nautilus species. So when you look at it in a little different way, probably a little easier, what we saw early on was what we saw in the oceans, and then after the first mass extinction, then we started seeing things evolving even more in our oceans, but even onto the land. And then as time goes along, you see where the extinctions have been marked. And everybody's friend, Tyrannosaurus rex, in the Jurassic period, was lost with the last mass extinction. That's when that mammals, et cetera, started really taking over on land. And this is not news to any of you who've seen this, and probably on Discovery or History Channel or whatever. But it's interesting because if you look at this in a context in regards to man, so we know the planet's at least 4 billion years old. How long have bacteria been here? Best guess from paleobiologists is probably about 3.5 billion years. They've been around a long time. And when you look to the left, you see it's filtered, it's probably close between gram-negative and gram-positive bacteria, but they've been around for about 3.5 to maybe 3.4 billion years ago. If you look at this whole timeline as a calendar year, they showed up around January 1st, maybe January 2nd of that year. It took until June of that year for fungi to come in, so about 1.7 billion years. When did we start seeing mammals? Well, December 24th on New Year's Eve, it took us that long before mammals came about because of all these extinctions. When did humans show up? We showed up the last day of the calendar year at 23 hours and 56 minutes. When did we start having antibiotics to use to manage a lot of these infections that we do almost on a daily basis? The absolute last day, 23 hours, 59 minutes, 59 and a half seconds. So they've been around about 80 years now. This is not even an eye blink. I mean, this is, if you look at the whole grand scheme of things, an extremely short period of time. Where did antibiotics come from? From an observation. Some were a little earlier than this, but the big one, penicillins, were actually, as you know, from Fleming's lab, and he was the one who made the observation that there was a mold on a plate and bacteria didn't like to grow up around it. And it was his curiosity that's like, why does that happen? What's going on with that? This mold was making a compound later known as penicillin. This is what you'll be receiving in your pharmacy now. It's the brand new generic version. No, I'm kidding. This is the old one that came out in an ampule. And you see it's a little discolored. It's not as pure as what we have today. So they really haven't been around too long. This was, as you remember, with the early 20s. We didn't have it commercially until the 40s. But these bacteria are really resilient. If you've ever been to Yosemite and to some of the national parks, including Yellowstone, I think Yellowstone was amazing. We went several years ago. This is the prismatic pond, and what do you see? All this red are bacterial mats. Here's a vent. This water is at least 190 up to 220 degrees, and some of these, if you go deeper, are superheated. And the bacteria that you see are alive and thriving on the edges of it. And as you see, there's a sign, do not mark bacterial mats, because people carve their initials into it, because we humans can't read signs. So they can adapt to almost anything. We've seen them growing in rock under high pressure, using things you would never think that bacteria would use. They use things like peroxide and sulfates. We have no idea how long it takes them to replicate, because we can't replicate this in a laboratory very easily. Having rock under high pressure, and then how do you know when the bacteria are actually growing? So some of this taking a year to maybe 300 years to replicate is conjecture. We also know that it grows at places that are difficult as well, and the under steam vent areas where you're under hundreds of thousands of pounds of pressure, but there's a hot vent coming up from usually a volcano or some kind of superheated water, and they thrive. And they're not using exactly the same compounds that they normally would, more towards the surface. We freeze bacteria all the time, right? You're doing research, you get a bug or a resistant one, something you want to do research with. What do? You put it into a bottle, put it in your -70 Revco, later on thawed out, and most of them will grow again. They're extremely resilient, or even in a desert under extremely low humidity, extremely hot temperatures, and you can find them there. So that's the point. They've adapted while they've been here for so long. And they can adapt to live in us, as you well know. This was, I thought, one of the best presentations I'd seen in a long time. It was one of the earlier presentations that Dr. Bonnie Bassler does. Bonnie has a lab that's in Princeton. And one of the areas that she looks at is actually quorum sensing of bacteria. Bacteria being able to talk to each other by chemicals. Have you heard of anything so crazy? We do it all the time in our cells. It's called hormones. So they know that there are compounds, and different species have different compounds that they can communicate with each other. Her area was because she was investigating a very small squid that lives in shallow areas out in Hawaii, and it uses bioluminescence to actually hunt. It has photophores on its back. It grows them in an organelle underneath and has little shutters to control the amount of light. So when it goes and it's hunting, it's really stealthy because it doesn't leave a shadow. But during the day, it's food, so it buries under the sand and gets rid of 95% of the bacteria, which are Vibrio fischeri, and they bioluminesce. There are several bacteria that can do this, but that's what this squid does. And she was interested because, does the light gradually come on or what happens? And she found that it doesn't happen that way. It gets to about 10 to the 9th to 10 to the 12th, I believe. And then all at once, the light comes on. That there are ways of communication that the bacteria know there's enough of them there, the light can come on, and that's how that they do quorum sensing. Staph aureus quorum senses, because if 100 of these gets into your body doesn't care. It's 100 Staph aureus. You make all the toxin you want. You're tiny compared to us. But if they grow to 10 to the 6th, 10 to the 7th, and they quorum sense, and now they make toxin, you can get overwhelmed. You can get septic. You could die. So this is a really interesting area for her. And one of the things that she, in her talk, and if you haven't seen it, one of the early ones was on TED.com back in February of 2009. She's done several. She does them now for Howard Hughes, the medical information, things that they put out. And her comment was, well, when I look out at an audience, usually people see people. She said, I see bacteria. Because we have about 1 trillion cells, but on us and in us, we have approximately 10 trillion microbes. Bacteria, fungi, etc. And what's really impressive is that because of the human gene project that we have approximately 30,000 genes. Well, from the bacteria living on us and in us, we have approximately 3 million genes. And remember, the bacteria scavenge. They'll scavenge for food, for proteins, for DNA, and they'll get plasmids that can jump from each other that are genetic. So the chance of variability is pretty significant. Well, we also want to know more about it, and that's why you've heard a lot about the Human Microbiome Project, wanting to know, because we all have these all on us and in us. And for the vast majority of the time, they're great, they're symbionts. They help us to stay alive. Our gut makes things that we don't normally make, some of the vitamins, etc. So looking at different areas, in the mouth, in the GI tract, in the stomach, or in the large and small intestines. In the urogenital area, especially the vaginal microbiome, is really interesting. And obviously on skin. So we're getting a lot of information about what are these things, what do they do, how do they protect us, how do they go bad, which I think is extremely interesting. So where did the title of this thing come from? There's a book that came out a few years ago, and that's the book, and it's called The Sixth Extinction. And I thought it was very interesting because it goes through, and that's all the different chapters that are in it. It goes back and it looks at how things happened in the past and some of the changes and some of the data. But realistically, I thought one of the more interesting ones was that one, the new PANGAEA. Because the concern is that we may be in the sixth extinction and in the new PANGAEA. The description in this chapter is that there's an evolutionary arms race in which each species must be equipped to defend itself against potential predators and need to be more fit than their competition, as it's kind of always been with us. But if we see, as a species, if we see something happen that we have no defense to, if it's a new fungus, a new virus, or a new bacteria, we may not survive. That's why we get concerned about pandemic flu and all these different things, because if we don't have a way of fighting this, a lot of us won't survive, and we would get into quote, an extinction. Not all of us may die, but a lot of us. So where do we fit in the new PANGAEA? And this is meant to just give you kind of a flavor of some of the things that are going on and the interesting things looking at this big area of research right now, which is the human microbiome. Most of you know the one quote at the top by Nietzsche, which is that does not destroy me makes me stronger. But frame it with Fleming's comment, not long after discovering penicillin and actually being able to give it to a few patients and then recollect the unmetabolized part of it in urine and then give that to people. That's true recycling. It's not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them. And the same thing has happened actually inside the body. We know this. We see this. We get consulted to see people who have pretty resistant disease going on or completely resistant disease going on, which is why I used to have dark hair. Now I don't. I used to have more of it than I used to. Now I don't. I'm just happy to have hair at this point in my life. So what I'm going to do is just to touch on kind of some of these 4 topics there is that adaptation, resistance, and virulence, new and emerging organisms, and some of the conditions that are associated with the microbiome. They may not be completely associated with microbiome, but it's touched in some ways. Some of the, what's going on with host resistance a little bit, and then what are the implications for the future, what's going on in the future. Well, the first is if you're looking at the gut, which is one of the biggest areas of research, is what's normal gut microbiota, and I will tell you without reading you a whole line on there, nobody really knows. Trying to quantify this has been a little bit more difficult than people can imagine because things do vary. They vary between countries somewhat. They vary among even in some states in the United States a little bit. Depends on your diet, depends regionally what are people eating and what are they doing. There have been 3 types of microbiota that are called enterotypes that have been proposed, but it's really tough to look at that in extremely young children because they just don't fit. So that's kind of still in flux as well. When you talk about microbiomes in the GI tract, they talk about these 4 phyla being the most common. There are a few others that will get mentioned, but these are the most common. And I know that you see the firmicutes, the Bacteroidetes, actinobacteria, and proteobacteria. So if I pick on somebody, you can give me a representative organism from each of these, right? No. I had to look them up too because we normally don't deal with this phyla at this point. So just to give you an idea, in the firmicutes, these are felt to be kind of the lower order, the earlier gram-positive bacteria, and they include Clostridia, Lactobacilli, Veillonella, Megasphaera, and Mycoplasma, as you remember, is somewhat cell wall deficient. In the actinobacteria, these are higher level gram-positives, and they include things like Mycobacteria, Corynebacteria, Nocardia, Rhodococcus, and some of the Streptomyces. If you look at the Bacteroidetes, these are gram-negatives and kind of older, and these are the ones like Flavobacteria, Chrysciobacteria, Capnocytophagia, Elizabeth Kingae, which, if you recall, there was an outbreak of that predominantly in nursing homes in Wisconsin. CDC investigated, and nobody can figure out where it came from, and it's gone, and nobody can figure out why. So these are really interesting when they happen. And the Bacteroides fit into that group as well. And then in the Proteobacteria, the things you know and you love, like E. coli, Salmonella, Vibrio, Helicobacter, Yersinia, Pseudomonas, and the Enterobacteriaceae. All these things that are in the gut tend to be in that area. So we know that a lot of these things can stick to mucosa. They hang around a long time. They protect us from other things, but then we have this other issue that it's not the same things in all areas. This is from one of the articles that I think I'd already referenced. Just looking at what's out there now in some of the publications, and you see that the same kinds of bacteria that we just talked about, if you look at the left bottom with the actinobacteria, firmicutes, etc., a lot of them are there, but just in different amounts, depending on what you're talking about in the GI tract. If you're talking about H. pylori negative stomach or an H. pylori positive stomach, gee, if it's an H. pylori positive, it's proteobacteria because that's where H. pylori lives in that phylum. So it depends on what's going on with individuals. And there's some data now that when you're born, you don't really have any gut bacteria. Your zygote doesn't divide into gut bacteria, at least not so far. We may do CRISPR and cause that to happen, who knows, but hopefully not. So you have to get it from someone or something. Usually it's acquired from mom during birth or shortly after birth. And you know there's a big thing now with C-sections, making sure that they get exposed to mom's microbiota so that hopefully you can keep those going forward. Well, this is data that just actually recently got published a couple of months ago. And they're looking at this whole concept, and in the past, our diets were different. Our exposure to these was different. So when you start looking at gut flora, it was probably more robust when you're talking about, a few hundred years ago as to what it is now that's over on this side. And this concept of a kind of depleted microbiota is very interesting because when you go back and look at the bottom with the innate and adaptive immunity, having seen a lot of these things, getting immune recognition for them is a big drive so that later on in life, if you get exposed, you've already seen it and it's no big deal and you're going to remember that with B and T cell immunity. Does the same thing happen on the right-hand side? And I think there's data that suggests it doesn't as well as it used to. That we have a little bit of loss of our adaptive immunity and looking at decreased tolerance, decreased reactivity, and perhaps being associated with autoimmune or other inflammatory things, or other allergic immunologic issues. And just to give you a flavor of some of those things, it's what happens with the bottom line here, which is dysbiosis. When you lose that kind of maximal what you're used to ratio, something comes in and causes a shift from one area to another. Most of what we'll talk about now is what happens with dysbiosis. This is from an article a few years ago from Nature Reviews Genetics. Now, this is not suggesting that every one of these diseases is caused by dysbiosis, but dysbiosis is felt to play a part in it. We're not sure exactly how it fits into each one of these, but when you look down and you're looking at psoriasis, there's a different ratio in people that have psoriasis of Firmicutes actinobacteria. So a little bit of different change in some of the gram-positives, interestingly enough. Obviously, reflux, you're looking at esophageal microbes, because you do swallow them all the time, also can change. And then lower abs and H. pylori. Obesity has been very interesting because there was a report of a patient who had recurrent C. diff, didn't respond to usual kind of things, and Venka with a taper, et cetera. So ended up having a fecal microbiota transplant. Well, the donor was the patient's mother and was obese. When the person got the FMT, it actually worked pretty well. They got rid of their C. diff, but then they started putting on weight. And they're thinking, and this has not been the only one. There have been some others, and looking at what bacteria might be useful for that. Because can you imagine, instead of having dietary pills for you and, looking at some of the naloxone-based things that are out there now, that you'll end up taking capsules, trying to repopulate your microbiota with the good ones so that you can lose weight. And of course, people joke about this all the time because they are out there for FMTs, that you can get FMT in a capsule, which they're affectionately called crapsules. And all kinds of, you can imagine, other jokes that go along with that. But I think when you look down through these, there is some data that suggests that what's in you, if there's dysbiosis, can affect other parts of you. Probably because of some of the signaling that goes on. And how does that interpret? We don't know, but we know it happens. A flavor with allergy. There's been a lot of studies looking at some of these, just a couple of things. In Scandinavia, there's been a few studies looking at low intestinal microbiota diversity in the first month of life. Has been associated with allergic sensitization to asthma in 6 to 7-year-old kids. We've seen that in other studies, that if you don't get exposed to antigens early in life, it does increase your risk of having asthma. There is an association. What happens with that, it remains unclear. In the UK, kids that are colonized as a newborn by particular strains of bifidobacteria, and some of these as young as a week. Later on, it depended on which species of bifidobacteria they got associated with atopic dermatitis. If they got Bifidobacterium breve, they didn't see it. But if they got colonized with Bifidobacterium I always have to stop, cannulatum, they had higher rates of atopic dermatitis. And there are others that I wouldn't say have probable but possible causations. This is an emerging field looking at, you are what you eat, or what happens to you is because of what you eat. Which begs the question for a lot of people is, have we lost some of this because that's what we used to do? That's probably what I lived in the upper left, was like mud. People used to ask me, what was your favorite toy when you were a kid? I said, dirt. Didn't matter. Army men, you played in the dirt. You had trucks, you played in the dirt. You were out playing Army, or you were playing, football, baseball. It didn't matter. You were in the dirt. You tripped and fell. You ate dirt. You were dirt. And now you're virtual dirt, because we're in homes where some people are obsessive about making sure everything is, quote, sterile or is high degree antisepsis, and we're taking some of those things out of them, like triclosan and other things that can be associated with other diseases. So it's been interesting to see is certain of this because of our activities. So we talked about C. diff. I don't want to belabor the point. Most of you already know pretty much what's on these slides with C. diff, that we used to see a lower toxin-producing strain. Now we have a hypervirulent strain that's around that caused us problems in Canada a few years ago. CDC, about a year and a half ago, released data that said there are about 500,000 cases a year of C. diff. 1 in 5 will have a recurrence, and 1 in 11 people at the age of 65 or over within a month after having this diagnosis. 80% of these people, 65 and older, would probably not be with us. They would die. If not directly from C. diff, from a C. diff-related complication. Data from, this is New England Journal back in 2015, looked at projections as to where we would be with C. diff because the trend has been positive. That's why you're hearing about Joint Commission looking at hospitals now and MDRO, multidrug resistant organisms, and they've now lumped C. diff into that. They're going to want to see when they survey your hospital, what are you doing trying to prevent C. diff? One of the driving forces with this is fluoroquinolone use, especially with the hypervirulent ones, because they're inherently resistant to quinolones. So if you're killing off everything else, leaving C. diff that can start replicating, there's no suppression to keep it from replicating and make toxin. And it's not just the little bit of toxin that it used to make, because those were the two, toxin A and toxin B, of the old strains. I just decided to put in so you can see why we don't always, it's like dermatology, they taught me this many, many years ago. You don't say seborrheic dermatitis, it's too difficult, sebderm, right? I mean, it's just easier. Can you imagine talking about binary North American pulse field type analysis 1, ribotype 27? We don't call it that, it's BINAP 1, 027, or most people just call it BINAP strain. This is the emergent, highly toxin-producing strain that killed lots of people in Toronto a few years ago. It wasn't that long ago. This has become one of the more common types that we see in the United States. You can test for this. We can test for it in our lab. If you have a Gene Expert machine, there is a cartridge that you can actually look for BINAP positivity in your C. diff strains. The problem is it makes huge amounts of toxin A and toxin B, and it also makes binary toxin that's encoded by these two genes, CDTA and CDTB. Those bind together, a huge other kind of toxin, and it's just nasty. These people generally tend to be significantly sick and have high white counts, as you know, elevated creatinines, etc. Risk factors, you know most of these. You've got an antibiotic, but it could just be advancing age. You get exposed to somebody out in the community that's got C. diff, and how does that happen? Like what we did when we were at our ID week last week. We decided to take a break one night and go see a movie. How many people sit in those seats, and they're cloth seats? How many of these people were having loose stool and had spores on them that you're sitting in? And now you're thinking about the seats you're sitting in. Who cleaned those the last time? Uncomfortable now? I was sitting in the seats in the convention center because hundreds of thousands of people sat in that. So I'm just wondering when I'm going to start having loose stool. And high severity of illness, but all these things can predispose you to some degree, even proton pump inhibitors, because C. diff generally would be susceptible to gastric acid, but of course when you put people on anything that's going to decrease gastric acid, the chance of them surviving making a small and large bowel is significant. So we know that the first thing to do if they're on antibiotics, if you can, stop the antibiotics. If you can't, then you're going to have to treat, and these are the three things that generally we look to, either vancomycin, metronidazole, or fidaxomicin. The problem is that we see, as you saw, recurrent disease. People relapse. It is interesting when you start looking at some of the data now that we've started looking at C. diff and the microbiome. Data suggested that the efficacy was better if we used vancomycin. We got away from it because of the concern about generating vancomycin-resistant enterococci, because you're giving a lot of vanco in a large bowel, and there's lots of enterococci there, and we started seeing that occurring, and it seemed to be related to the use of oral vancomycin. We went to metronidazole, but the efficacy is not quite as good, especially not for these highly aggressive binary toxin-producing strains. But what people weren't really looking at is, what does vanco do to your gut flora? It's not benign, as a matter of fact. If you look at 3 drugs that we just talked about, vanco, metronidazole, and fidaxomicin, vanco changes the gut flora and actually makes it a little bit more difficult to get rid of C. diff recolonization, and call that colonization resistance. This can, it facilitates anywhere from 3 to at least over 2 weeks, maybe even a little longer, after you stop the vanco, of getting colonic flora back to where it was. You don't see that with metronidazole. You see less of it, I'll put it that way, and you see significantly less of it with fidaxomicin. This has been looked at in murine models, and giving them this species 24/7 in Bacteroidetes taxa, they couldn't find it. And this is normal, what's in a mouse microbiome in their gut. An overgrowth of Enterococcus proteobacteria and some lactobacillus and Clostridium. So they took longer to repopulate with normal gut flora after vanco. We saw the same things in treating bacterial vaginosis with clindamycin or metronidazole. Actually, metronidazole was very selective in getting rid of some of these anaerobes. Whereas you gave clindamycin, Clinda was like just doing napalm. You're trying to get rid of the weeds, and you hope that the weeds don't grow back and you get the trees and the flowers. So this is something that's been very interesting. As people are talking about, because you get dinged if you have C. diff that occurs while they're in the hospital, but they may be colonized before they get in there. People have started screening and then thinking about giving them prophylactic vancomycin. I think that might be a bad idea. We don't have enough data to go to that at this point. As a matter of fact, the VA is doing, and we're going to be part of it, of a 3-arm study, because no one will ever do this, of first and second recurrence with C. diff, looking at vanco with a taper, or fidaxomicin, and see if there's any difference in recurrence rates. This is an interesting concept, and there's some papers that are coming out. The C. diff in the gut. Can your microbiome rejuvenation keep you young? An interesting thought. Why did this come about? Well, because of C. diff patients who have more severe disease tend to be older, as you just saw from the CDC data. So there's a very interesting murine mouse model where they looked at older mice versus younger mice. They gave the older mice C. diff and younger mice C. diff. The younger ones did actually really well with it. No mortality. Older mice did not do well, and there was some mortality. And when they looked at them, they saw that the serum levels of anti-CDF toxin A and IgM, IgA, and IgG were significantly lower, and they had an altered microbiota composition that was much different than the younger mice, and that everything was lower when you looked at older mice versus younger mice. Their pro-inflammatory cytokine expression, neutrophil recruitment, and tissue damage was much worse than older mice. Then they did an interesting thing that we like to do in research. They did a crossover. So they took the young mice who had been in their cages, and the old mice had been in their cages. They put the old mice in the younger mice's cage, because mice sometimes, not uncommonly, are coprophagic. They ate the young stool, their microbiome. They did that for a couple of weeks, and then re-challenged them with C. diff. They did really well this time, that they didn't have any deaths, and their pro-inflammatory cytokines were significantly higher. And actually, they weren't the ones who had seen C. diff before. These were different mice. So this begs kind of the question is, can we rejuvenate our microbiome by ingesting a younger microbiome? There's also data out there that, and you probably have seen this, of people who got young people's transfusions actually, if they were older, significantly increased their immune system as well. So for those of you who are young in this room, we're going to start farming you and harvesting your blood and your stool, and because we want to stay alive. It's just the cost of business. Any of you know about Ponce de Leon? What did he do? Especially for Florida. What did he say he discovered? The fountain of youth. And where's the fountain of youth in Florida? It's in St. Augustine. You go and it's got this big arch, and they have this big fountain with water coming out of it. I would suggest to you that's not the right place to go. Because realistically, I think we're going to start doing this. Now, no, this is chocolate. And I'm not saying anything else, but we kind of do that with our FMTs. But FMT for everyone. This will happen at the Christmas party, right? No. I hope not. Another area that people don't think about is, you saw Elon Musk just said we're going to get rid of all the Dragon capsules, new capsules to go to Mars. What do we do with our microbiome if we're going to go to Mars and live on Mars? Because we're protected by our magnetosphere. We get extremely limited amounts of radiation, and you get outside of that, radiation goes up significantly. They're not sure exactly what they're going to use to protect ships, but actually the best thing is to coat them in a layer of water. Same thing you do in nuclear reactors. It's better than lead, because if you have a gamma particle hit lead, you're going to get other high radiation come off of that. Not the best thing to do. So we don't know. They've tried looking at this in flight, but astronauts are not microbiologists. The tests were very rudimentary. They got different results. So one of the things they decided to do a couple of years ago was to send the twins, who were both astronauts, Scott and Mark Kelly. Scott went up, stayed for a year. They collected samples, looked at his microbiota while he was in space, compared it to his brother, and they're not exactly the same. No person's exactly the same. It was interesting. When Scott was in space, his microbiome changed. He had more firmicutes that changed over to Bacteroides, but when he came back to Earth, he went back to what he was doing. How much of this is what they're eating when they're in space? It's not the same as they eat here. The other thing that was interesting was when he was in space, his telomeres got longer. But when he came back, they shortened. Will we live longer in space? I don't think so, because the radiation is significantly higher. That does things to your microbiome. There was a really cool space experiment where they tried looking at salmonella. They got it off of the returning space station capsule that came down within 3 hours, played it out, and there were some significant changes, more pathogenicity. The other thing is antibiotics in space, and this is a list of what they keep on the International Space Station. You see lots of different things, a couple of combination ones. How well do they stand up in space over the long haul? Because we probably are not going to be manufacturing antibiotics, and some of these are okay, but when they've tested some of these, especially the combinations like amoxiclav and sulfamethoxazole combinations, do not dissolve well, and they are not useful after they've been there for periods of time. And I think it depends on what period of time are we going to look at. We're talking months to years. So we need a lot more information about how do we keep our gut biome healthy if we're going to do space travel. To get to the ridiculous, remember I talked about oral vanco and vanco resistance. People had noticed that there were some strains of enterococci that became vanco-dependent. They've been living in low levels of vancomycin for periods of time. They were using it as food. You saw a few of these, but this has been back in 1999. 5 BMT patients up at Hopkins ended up getting this. 3 out of the 5 were demonstrated to be all exactly the same and vancodependent. People were on vancomycin for at least 3 and almost 4 weeks. That's why we don't use that in our BMT anymore. If you don't have a line or something, you don't prophylax for gram-positive infections. So when they quit using vanco and infection control things, that went away. But it's just amazing when you look. This is a plate on the left, and the one that's got all the stuff around it is vancomycin. And these are the VDEs, the vancomycin-dependent enterococci. Here's an E-test on this side. If you look on the left side of the E-test, you see all these clusters of bacteria, lots more at the top. That's where the antibiotic concentrations are higher, less on the bottom. These are vanco-susceptible enterococci. And you see that they're inhibited by high growth, less with you. It's completely, this is crazy. We're feeding them, and they're eating vancomycin. We did this. This is nuts. So that's why stewardship is a big issue at this point. We also know with pneumococcal vaccines, and I won't go through them, we've evolved 2 different kinds of them now, and they tend to be very effective. We know that some are more aggressive than others. If you would compare them to serotype 14 out of these multiple different kinds, some are vaccine and some are not, that types 3, 6A, 6B, 9A, and 19F are a little bit more aggressive. 1, 7F, and 18 are a little less from this article. And my mentor in medical school, Murray Musson, had this arrangement with the 3 hospitals in town. Every time they had an isolate of strep pneumo, they sent it to him, and he typed them. And they had history of what was going on. Published this in December, which is extremely interesting, as vaccines came out, they saw less and less of these strains. They had 193 isolates from children and over 1,000 from adults. The case fatality rate in adults was about 19%, and only 2 children died over that period of time, which is great. You know that the rate of mortality from pneumococci hasn't changed since Austrian and Gold's initial paper back in the 60s. It really hasn't, despite newer, better antibiotics. Nothing has changed. What they saw was as we closed the niche for these vaccine-susceptible ones, others started to move in, causing disease. And some of these non-vaccine types, but some of the vaccine types have even evolved to causing disease. So there are newer pneumococcal vaccines in the pipeline. We worry about pertussis. It seemed to happen after we quit using whole cell pertussis vaccine, and we went to acellular because arms hurt too much when people were doing this. So we thought this would last for at least 10 years. We're finding out now that DTaP lasts maybe, if you're lucky, for 4 years. And that's why we're using more frequent immunizations. And just to give you an idea, this is a really nice article Stan Plotkin did a few years ago now, is what can we do to get a better pertussis vaccine? All these kind of recommendations, which would be okay, but you see a lot of these are going to need trials, it needs time. Can we just keep re-immunizing, which is what we're doing? This whole cocooning thing of trying to immunize people around a small child is kind of gone because we're really trying to immunize pregnant women every time they're pregnant. One of the things I think is interesting is, these bacteria have adapted because one of the surface antigens that's in the vaccine is called protactin, and it's pretty immunogenic. From an outbreak a few years ago in Australia, what you see is this big jump, and these are protactin deficient pertussis. Bordetella pertussis with no protactin. So you've lost that site. Even though you have great immunity, it doesn't do you any good because that antigen is no longer there. Some unusual therapies in bugs. This is a big one that's up and coming. Another one's up and coming I won't tell you, I'll just mention to you because you'll see it in the media, is Candida auris, a highly resistant candida that can be misidentified, that the automated systems are not really good. The recommendations from CDC as of last week are if you get a Candida species and they don't seem to be doing better, you have to use MALDI-TOF to identify it. You have to do basically mass spec to identify it. Lots of hospitals now have MALDI-TOF systems. And you clean the room like a C. diff patient. You're using bleach or peroxide to clean the rooms because this thing can be everywhere. It can colonize rooms for at least 9 months, if not more, and patients, and be on their skin. We've never seen a fungus do this before. With this, just suffice it to say from time that we're seeing a lot of these hypervirulent Klebsiella, they're really sticky. They make a lot of slime, and that's part of why you see this. Even causing necrotizing fasciitis. Single agent, single bug causing neck fas. We don't see that here, but they've seen it in China. And then they've started seeing this now with a hyperviscous necrotizing fasciitis. The biggest thing that just happened with this is carbapenemase production. High-level resistance. When this happened in a hospital recently that you saw was in Lancet Infectious Disease just in August, these people, 5 that had trauma, ended up on a vent, got this in their lungs, and there's nothing really to treat it with, and they died. So as this starts moving around the world, we're really, really concerned. Unusual therapies, just to mention to you that people with recurrent UTIs, a lot of these are not really UTIs, it's asymptomatic bacteriuria because urine is not necessarily sterile. People can get colonized with bacteria, especially if they've had Foley's long-term devices, etc. Stones get infected, like struvite stones, etc. You can't get antibiotics into those stones, and unless you get rid of the stones, that infection is always there, or at least they're colonized. So this was novel because we learned a lot about that, and there was one strain that from these asymptomatic bacteriuria patients that's not pathogenic because of all the differences that you see listed here. So this 13972 strain, ABU strain, has been used therapeutically in 20 patients who had recurrent UTIs to E. coli. So what they did was colonize them with a non-pathogenic strain. And this is kind of what you saw, and they did it placebo-controlled, and these were the outcomes. That after 1 to 2 to sometimes 3 administrations till it took hold, and over, as you see, almost up to a year, significantly less symptomatic UTIs with that preparation than with placebo. Will we be using bacteria as therapy? Maybe. The last was this horrible one of using phage therapy of the patient's condition. These are viruses for bacteria. And what we see with this is that we used to use this before antibiotics came around, but we didn't know how to use them effectively, so antibiotics were much easier and people don't do this anymore. Of interest, there are a couple of companies that have phage preparations that target listeria, and you see it's called ListShield and Listex. They've been FDA licensed since 2006 to treat meats and cheese to try and prevent listeria. You heard about this? I didn't either until I started looking into this. What are they doing in food industry trying to keep us healthy using phage therapy? It's interesting. This is an unfortunate gentleman, 69-year-old professor, was in San Diego, went traveling, went to Egypt. Late 2015, while he was there, got pancreatitis, unfortunately developed a pseudocyst, was not doing well. They drained it, but they put a catheter in it, and then it got colonized with multidrug-resistant Acinetobacter, only susceptible to very few antibiotics. They tried treating him. He's not doing well. And they finally got him medevaced back to San Diego, where he lived with his wife, who's an ID practitioner, because he is not doing well. His catheter got dislodged, and then he got peritonitis with this MDR Acinetobacter. This is premortal. He didn't do well. He got comatose. The person you see on the left of that picture is Dr. Chip Schooley, who runs ID there. They got emergency use from the FDA to use phage therapy because the Navy keeps a huge bank of phages, and there's an upstart company that's looking at this as well. So they're using combination of phages against Acinetobacter, as well as continuing some antibiotics. And as you see, they treated him, and it took a while. It took several weeks, but as of June 6th, it's gone, and he needed extensive rehab because of the deconditioning, as you can imagine, being comatose for a period of time. He finally left the hospital in August. So we're using phage therapy. World's running out of antibiotics. I don't have time to go through all these, but suffice it to say that there are novel things coming about. Monoclonal antibodies, one of which you just saw that got FDA released for C. diff, vaccines, probiotics, relevance inhibitors, and this is the thing that, I don't know if it's good or not. Even DARPA, DOD is looking at this, and their project is called the Pathogen Predator Program, of creating bacteria that will eat other bacteria. And we hope it doesn't turn into a terminator bacteria because then it might start eating us worse than, I don't know. So we are in the new PANGAEA. We're trying to increase our odds. My recommendation is to do what we've always done, is to recognize these threats, get creative, and as one of my colleagues knows very well, how to persevere. So perseverance is what's going to keep us on this planet because the threats are out there. The threats are always changing. There's always new things. That's why it's so cool to be an ID, but difficult. There's always something new. And with that, I'm going to stop. And thank you very much for your time.
Is there any information that any of these either contribute to or defend against colon cancer?
It's very interesting because, as you saw, there are some of these anaerobes that are felt to play a role in that. Is that the only thing? And I think maybe not, but some of these, even some of the E. coli strains are felt to be associated with inflammatory bowel disease in some degree. I think if we can identify that, and that may be a role for some of these probiotics, people are looking at that. One of the things I was astounded with at the meeting was in it was the Netherlands, where everybody goes into an ICU. Doesn't matter what you've got. You go into an ICU, they give you either a paste or a slurry that has colistin, amikacin, and amphotericin. And for 4 days of ceftriaxone IV across the board. And they say they don't see any resistant organisms or anything else. And I said, what about C. diff? We don't see it. I couldn't see that. And they're looking at the microbiome, which to me sounds crazy. Because there is, and we actually have one that we inherited from another hospital, an MCR1 gene, E. coli. It's an ESBL-producing E. coli. MCR1 encodes for colistin resistance, which is emerging and going around the world. And CDC has a surveillance team looking at that. We know it's expressing it because we grew it and it's colistin resistant. What do you do for it? You keep them off of antibiotics. You just, and it's contact precautions, unfortunately, because it's a tier 1 organism. It's an evolving resistance. We don't know enough about it to know what to do with it yet. So we're hoping that he'll colonize and send his urine. So he has a biofilm or something. What do I do with it? Do I get it with this ABU strain and try and give it to him? That'll be 6 months in an IRB, easy. I don't even know where to get the strain. I don't know if that would work. There's all kinds of things to do, but probably the best is leave him alone. Don't give him antibiotics unless you really need to. And hopefully it'll repopulate with something else, or it will just go away. Yes, and I don't know how much time we have, but.
Can you comment on room sterilization procedures? Because there was that recent study that showed if you were in a bed or a patient,
Oh, yeah.
you did
Sure. Well, think of how I felt in this whole auditorium with all these cloth seats you can't really treat. It's an issue, and I will tell you that trying to treat that is going to be difficult because it's not only what's on the bed, but it's how well do we clean under the beds and everything else. And that's why that you've seen this emergence of two